So then the next speaker will be Dr. Eulberg. He's from the company NOXXON Pharma, and maybe one of you have already attended yesterday also talk about more general the platform of the Spiegelmers, which are developed by NOXXON. I'm very curious to hear today, at least that was promised yesterday, that we hear today an update about the clinical program. So the floor is yours.
Okay, thank you. You can hear me. Thank you very much for having this opportunity to speak today. I will talk about some highlights of the development of our clinical candidates. They are all so-called Spiegelmers, and Spiegel is a German word for mirror, as many of or some of you may know, and they will shortly also know why. I'll talk about 3 different candidates that we have in clinical development. So a little bit about the platform. The Spiegelmers, they are injectable oligonucleotides. They are short pieces of RNA, and they are not used as a kind of genetic material like siRNA. We use the oligonucleotide as a scaffold to bind 3-dimensional target structures. In order to, they work in peripheral blood, so in order to counteract quick renal elimination, they are conjugated with a 40 kilodalton peg. So far, we have some clinical experience with more than 300 subjects dosed, and all 3 substances were good, tolerated, and quite safe. Why do we use, I haven't mentioned that, they are not normal pieces of RNA, but they are mirror image pieces of RNA. They are L-RNA. The ribose moiety is in the L configuration. And why do we do that? Because such an L-RNA is biologically stable. It's just not a substrate for nucleases. And due to the same mechanism, it doesn't activate toll-like receptors. We produce that with a process called SELEX, where you have a combinatorial library of many different molecules. 10 to the 15 is a typical number. And by starting that with an already mirror-imaged target made on non-natural amino acids, a D protein, we get an aptamer to this D protein, and if we now just use the sequence of the identified RNA, and we synthesize that from non-natural L ribose building blocks, we get to the so-called Spiegelmer that binds the natural target, which is biostable. So we think these molecules are particularly suited to target chemokines, because chemokines, they have 2 different domains. Chemokines are important for trafficking of immune cells throughout the body. One of the domains is keeping the chemokine on the surface, because they are kind of signals on surfaces, for example, of the endothelium. And the second one is the receptor binding. And we could show, here you can see a crystal structure of a Spiegelmer with its protein, that the interaction surface is quite large and covers these both functionalities. Now we come to the first Spiegelmer, NOX A12. The target is CXCL12, or also called SDF1. This is an important communicator within tumors, between tumor cells and the microenvironment, which contain many immune cells, fibroblasts, and endothelial cells, and other types of that. And it has been shown that this target in cancer can reduce tumor cell death, promote proliferation, promote formation of new vessel and metastases, and by stopping that pathway, we think we can do a lot to stop cancer growth. In principle, there are 3 possibilities where this target plays a role in cancer. One is some tumors protect themselves by producing this target, not letting in immune cells, like cytotoxic T cells. The second is to block the tumor repair. A tumor that is, for example, irradiated will call for help, and bone marrow cells will come to start angiogenesis again, and this will then lead to regrowth of the tumor. You can interact to that. That's something we're going to do with glioblastoma. And some data I will show is the 3rd pathway. You can expose hidden tumor cells in case of blood malignancies and make them accessible to chemotherapy. That's what CXCL12 does in multiple myeloma. Here in the bone marrow, the CXCL12, the yellow ball, is produced, and the MM cells, they home towards the bone marrow where they are protected. If you inactivate the CXCL12 with our molecule, these cells are liberated and can be targeted by therapies. And this has been shown here. That's an animal model, a mouse model. Our drug only mobilizes the cells. It doesn't kill them. So we need to combine with a killing agent, which is bortezomib, or Velcade in that case. And here you can see this is the effect of placebo. This is our drug alone. Just mobilizes, doesn't kill them. And this is the killing agent, bortezomib. And if we combine that with ours, the response is deeper. And this is also shown here at week 5. So the next step was to really bring that to the clinic. This is a clinical study performed with 28 multiple myeloma patients. And this is the overall response rate. And of course, this was first exploratory study. There is no control group, but it is quite obvious if you compare that with historical controls that have a response rate of approximately 40%, that there is an improvement. Very important, we did not see addition of relevant side effects, so the drug was safe, and we are preparing a pivotal study with this drug in this indication. The second drug, called NOX E36, it targets another chemokine, CCL2. And CCL2 is a link between diabetes and the renal inflammation. In diabetes, you have high blood sugar, you have high blood pressure, and all that leads to a raise of our target, CCL2. Now, CCL2 does a lot within the kidney, where some cells carry the receptor, and that leads then to some unwanted effects, for example, inflammatory infiltration, and in the end you have a mildly inflamed liver, glomerulosclerosis, which means that the glomeruli will scar and they will die, and patients will lose filtration rate, and on the other side the filtration barrier breaks down, and the proteinuria gets through and into the urine, proteinuria. So that's what you see in many diabetic patients after a diabetic career of maybe 15 to 20 years. This is preclinical data to show that. Here, first of all, you can see that the influx of inflammatory cells is reduced, with vehicle here on the left, less leukocytes if you treat that with our Spiegelmer. There are regenerative processes triggered, so podocytes, which are important to keep the barrier intact, the filtration barrier, their numbers are preserved. And finally, you see that the glomerulus structure is maintained. So this is a healthy glomerulus. This is already one filled with fibrous tissue. And this leads to functional loss in the end. And this is then also translated here in the functional parameters, whereas the vehicle-treated animals have lost filtration rate. It is preserved in the treated animals, and the albuminuria was also improved. So the Spiegelmer appeared to have triggered some structural changes that result in improved kidney function. And this preclinical data have also been tested in the clinic in a 75-patient study, and we could see principally the same in terms of ACR. So there is a reduction in proteinuria. Of course, glomerular filtration rate in humans progresses so long that you would need to have studies of 1 or 2 years of duration, because otherwise nothing can be seen. And the important thing is that these beneficial effects, this is the cessation of treatment, that this was maintained even for 3 months of follow-up, which was quite astonishing. But it is a hint that there is structural change triggered by this treatment. If you then look at that patient population that we will use in the next study, where certain medications will be forbidden, where nephritic syndrome, obvious inflammation of the kidney that is not related to diabetes will be excluded. If you look at that, that was a post-hoc analysis, of course, but then the effect was even much larger in this subgroup of patients, which was 2/3 of all included patients. So, third molecule, that is NOX H94. Target is hepcidin, and hepcidin is a key regulator of iron in the body. Hepcidin is an acute phase protein. It's upregulated when inflammation is kicking in, and hepcidin works on this ferroportin, which is the iron exporter from iron-storing cells. We have these cells in liver, in gut, in spleen. And if hepcidin is upregulated, ferroportin is closed. No more iron comes out of this storage. The plasma pool of iron is depleted, and of course there is no more iron for use by the bone marrow to produce new erythrocytes. So in chronic inflammation, patients tend to become anemic, and hepcidin is one important player there. So we wanted to show that in a pharmacological model with NOX H94. This is a monkey model. This was necessary because the molecule is only active in the monkey, not in rodents. And these monkeys, they get IL-6 to get an inflammatory stimulus for some days. And then you can see a mild anemia setting in. Day 4, day 6, about 1 millimole per liter reduction. And if we treat them with our drug, this is reduced. This is even more clear when you look at the reticulocytes, which are the first cells that profit from more availability of iron, and where it's highly significant. The same could be shown also in humans. You can do the same in humans with lipopolysaccharide, and we could show that iron is liberated. You can't show that the anemia is produced because that's not feasible in humans, but the pharmacodynamics of inhibiting hepcidin to improve iron availability could be shown here. And next studies will then be studies in patients. To sum up, Spiegelmers, the platform, it enables generation of tailor-made therapeutic molecules. We get a target, and we made a binder for that. They are suited to target chemokines. I told you about 2 of our candidates that target these communicators between leukocytes and body cells. 3 Spiegelmers have been in the clinics today, and they have all been thoroughly characterized in full preclinical pharmacology and safety. They can be produced by a solid phase synthesis process, scalable GMP compliant process. They have been administered to a large number of subjects, and we have done a series of exploratory phase 2A studies where we have seen clear hints of efficacy. So how to get on has already been discussed with regulatory authorities, both here in Europe and in the US, and we are preparing pivotal clinical studies. So I hope we have left childhood and starting to become of age with that substance class. Thank you very much.
Any questions?
Thanks. Quite an interesting concept, but I may have missed something. What is the advantage of this technology when you compare it, for instance, with other technologies that interact with chemokines or pharmacologically active compounds? Take, for instance, monoclonal antibodies. What is the advantage of your technology?
Yeah, you saw the second slide with this crystal structure. Chemokines have these 2 domains. One domain is called the heparin binding site, which is a quite positively charged stretch of the molecule. And with this side, it sticks to the endothelium. For example, a chemokine is produced deep in the tissue where there is an infection, and the chemokine gets to the endothelium from inside the tissue, and it has to stick here. It's a danger signal to call leukocytes in. So we have shown that our inhibitor NOX E36 interacts with 5 of the 6 crucial residues that are important for this function. And then there is another domain that these chemokines have, which activates the receptor. And for this, the interaction surface is so large that this also encompasses these 2 out of 5 crucial residues that are important for receptor activation.
So the affinity is much higher than for, let's say, monoclonal antibodies or small molecules that you use?
Well, the affinity is actually not an issue because at the concentrations you bring that into the blood, the normal concentrations you can achieve is in the micromolar or hundreds of nanomolar range, but they are sub-nanomolar.
But I noticed that you use PEG to
yes.
Improve the circulation time, I guess?
Yes.
And that doesn't affect the affinity for the chemokine?
Yes, it can. So every Spiegelmer that we identify is individual, absolutely, depending on the fold. And we have had cases of during drug discovery where some of them were just unfunctional when you coupled that with PEG. That's trial and error. Luckily, you don't only get one molecule out of such a selection process. You have more to pick, but you have to try.
Very interesting talk. I have a question. One issue with the CCL2 inhibition, in I think particularly in human, has been a huge accumulation of CCL2 inhibitor complexes, which has actually probably led to the fact that you have a residual free CCL2 which you cannot avoid in tissues, counteracting the inhibition. So have you looked at that in your case?
Yeah, actually that's correct, in particular because we have a molecule that has a total mass of 54 kilodaltons, and it binds to the chemokine, and the complex is circulating. This is correct. We have shown that this circulating complex, as it is in an animal or in a patient, does not contain free target that can induce chemotaxis. That has been shown. The Spiegelmers are degraded by uptake into macrophages, or generally into phagocytes. And we think that the Spiegelmer together with the target are then incorporated then where they are degraded in this compartment.
So then let's thank the speaker once again. And let's go forward then to